bioinformatic approach based on the LC-HRMS data that had been obtained. First, first eye extraction of temulawak for 24 hours and 4 times the solvent replacement at room temperature, and then analyze of active compound using LC-HRMS, and then tabulation of active compound data. And then we did molecular docking process with start preparation of ligand and receptor until visualization. This is until visualization of molecular docking result. And this is in the study predict this is the result of molecular docking, and in this study predict that 3 active compound, curcumin, artemisinin, curcumin of curcuma santoriza as potential agent in inhibiting MAPK8 protein. And the molecular docking results showed that MAPK8 protein inhibitor disease had the highest binding affinity value of minus 9.0 kilocalorie per mole. Minus 9.1 kilocalorie per mole compared to 3 compound of curcuma santoriza. And then, however, the binding affinity value of curcumin, this is curcumin, is close to the control of minus 7.4 kilocalorie per mole. And then previous study suggests that binding affinity value that more negative show ligand receptor interaction is greater. This is sorry, okay, 4 minutes. And then visualization results showed that curcumin, artemisinin, and curcumin have strong interaction as inhibition of the MAPK8 protein, indicated by 3 compound of curcuma santoriza had the same active site compared to MAPK inhibitor. This is a visualization of PMOL, and then okay. And we now show that all active compound have the same active position with MAPK8 protein inhibitor. This is yaitu MAPK inhibitor. This is curcumin, and then this is artemisinin. This is finally curcumin. And then the amino acid residues that bind through this is hydrogen and hydrophobic interaction, and amino acid residues form on the ligand protein complex, including lysine 55, valine 140, methionine 108, methionine 111, glycine 33, alanine 53, and leucine 168. And then the same amino acid residue with various interaction on MAPK8 ligands complex have several functions, such as increasing the binding affinity value and play an important role in stabilizing the bond between ligand protein. And then previous study have showed that curcuma santoriza function as an anti-cancer agent. Okay, and this is analysis of the protein network by string show that certain protein directly interact with the MAPK8 protein in regulating the apoptosis process. And then this is previous study show that MFPK can regulate apoptosis through specific phosphorylation of downstream mediator of apoptosis, including the tumor suppressor P53. This is in the conclusion. In this study, R1 active compound in curcuma santoriza predict have potential as anti-cancer agent indicated by the binding affinity value close to the control and the same binding set. And then in vitro and in vivo studies, we needed to validate the anti-cancer activity of curcuma santoriza in inhibiting MAPK8 protein. Okay, thank you for your opportunity. Wassalamualaikum warahmatullahi wabarakatuh, and then good afternoon.
Afternoon, Ibu Nur Fitriana. Very interesting topic. Now, can we move to the second presenter?
Okay.
Will be Ibu Suni Sofia Anika. Ibu Suni Sofia Anika, are you there?
Yeah, I'm here. Can you hear me?
Sure, I can. So,
may I share my screen? Hold
sure.
on. Okay, wait a minute.
Okay, Ibu Sunia, time is yours, Ibu.
Okay, thank you.
Welcome.
Can you see my screen now?
Not yet, Ibu.
Not yet.
Okay. Okay, I'm sorry for the delay. Hold on.
It's already in my screen, Ibu.
Is it the slideshow? Already the slideshow?
Yes.
Yes, already, Miss.
Okay, thank you. Okay, good afternoon, everyone. Thank you to the committee for this opportunity for me to join the International Biology Conference together with all other great participants. We are Suni Sofia Anika and Jumailatu Soliha from Biology Study Program, Faculty of Science and Technology, Limjunan Kalijaga, Yogyakarta. So today we'd like to present our research with the title Expression of MTP1 and HMA3 genes in Amaranthus viridis stressed with zinc. Okay, so the species that we used for this study is Amaranthus viridis, which is one of the plants that is commonly consumed by our society, especially the people from Asia. So this is the plant that people used to consume as food, where we use it in food and where we use it in our cooking, as well as in traditional medication treatment. So one of the criteria why this species is commonly consumed is because it grows well in most environmental conditions. However, this species is known to have a potential as a hyperaccumulative plant. So this plant is tolerant to high concentration of heavy metal. One of the heavy metals that this species is tolerant to is zinc. So it is able to accumulate and translocate high amounts of zinc in the upper parts of the plant, which is the leaves and the stem, which are the parts that we use to consume in our food. So through the consumption of this species, there is a possibility that the zinc inside the plant could be transferred to human body, and this will then possibly could cause health problem if the concentration of the zinc is higher than the limit. Okay, then. So based on the ability of these plants to hyperaccumulate heavy metal in high concentration, it is because of the presence of transporter protein that cause the plants to be tolerant to the heavy metal. So one of the transporter proteins that we expected to be present in the plant is MTP1 and HMA3 transporter proteins, which are synthesized by the genes, which are the MTP1 and HMA3 genes. So in this study, we tried to identify the presence of this gene so that we could know if the protein is present or not in this plant. So from this information about the protein present in this plant, it could provide information for further study and improve understanding on the translocation and transportation of zinc, so that in the future, it might be useful for consideration to prevent health problems. So this is, hold on. Next is the material and methods. Okay, so these are all the steps that we used for our research. So basically, we started with the growing of the plants and the treatment of zinc on the plant. Then from the plants that we treated with zinc, we isolated the total RNA from the plant sample. Then we conduct RT-PCR, reverse transcriptation polymerase chain reaction from the RNA isolated using the primer that we designed on our own. Then to visualize the results of the PCR, we use electrophoresis. Then from all of the observations and the results, we did the data analysis from it. So for seedling preparation, we identified the species using the identification keys. We collected the seeds from it, then we did the sowing. After that, so we give the treatment, zinc treatment to the zinc by mixing the growing media with a zinc chloride solution. So we use 4 different concentration of zinc on the growing media. And for each concentration, there are polybags with the presence of plant, and 1 polybag without the presence of plant for each concentration. Then we grow all of the plants in the polybags. So on the first day, we moved the seedlings to the polybags. And since the first day of the zinc treatment on the seedlings, we did the measurements and the observations on the plant until the last day of the zinc treatment on the plant. Then on the last day of the zinc treatment, which is after 21 days after the first day of the planting, we selected only two plants from all of the samples. First of all is the control plant with the best growth amongst other control plants. And the second one is the plant treated with the highest zinc concentration and with the best growth. So in this case, the one that grew well was the one planted from the highest concentration, which was 1000 ppm. And from these two plants, we picked two plant parts, which are the leaf and the root. So these leaf and root from each plant was brought to next step, which was the laboratory steps, which is the total RNA isolation. So before the PCR, we designed the primer to be used for the PCR. Then for the PCR, we used the RT-PCR. We synthesized the cDNA from the total RNA isolated. Then we did the amplification. Then to see the results of the PCR, we use electrophoresis method. Then for the data analysis, we did qualitative and quantitative analysis. First of all, the qualitative, the appearance of the plant parts, such as the change of color and so on. Then the next one is we observe the presence and the appearance of the amplicons after the PCR step. Then quantitatively, we did one-way ANOVA analysis to see how the zinc concentration in the soil influence the growth of the plants. Then we also measured the length of the amplicons formed on the electrophoresis gel.
Pardon, Ibu Suni, you only have 1 minute left. So could you speed up your presentation?
Okay, I'm sorry. Okay, so for the results and discussions, so to simplify the results and discussions, so we will just go directly to the expression of the genes. So from the expression of the first gene that we studied is expression of MTP1 gene. So we identified that from the electrophoresis gel, we found that there were 4 MTP1 amplicons appeared from all of the samples. So from all of the samples chosen, all of the amplicons appeared. And the level of that expression were visually similar from the leaf and the roots and from the both plants with and without the zinc stress. And that's for the MTP1. Then next is for the HMA3 expression. So on the other hand, the HMA3 gene, the specific amplicon wasn't, they were instead, some amplicons were formed on the gel. And all of the amplicons formed were from the samples, were only from the control samples and not from the samples that were stressed with zinc. So this was expected to be, the reason was expected to be because of the non-specific primers that were used. Next, this is the visualization of the electrophoresis for the expression of both genes. The left one is for the MTP1 genes, and for the right one is for the HMA3 genes. Okay, this is the visualization of the result. Then lastly, the conclusion is MTP1 gene is expressed in the leaf and root part of Amaranthus viridis and was not influenced by the zinc concentration in the growth media. And the amplification produced amplicons with the length 79 base pairs with a visually similar level of expression. On the other hand, the expression of HMA3 gene couldn't be concluded yet in this study because there was no specific amplicons obtained during this study. I guess that's all from me. Thank you so much for your attention. I am going to give the moment back to the moderator. Thank you.
You're welcome, Ibu Suni. Thank you very much for your very detailed presentation. Now we will go to the third presenter. That will be Ibu Yatri Hapsari. Hello, Ibu Yatri.
Hello.
Hello, can you hear me, Ibu? Yeah. And are you ready to give us the next presentation for this moment? Yeah. Are you ready to deliver your presentation?
Yes. Okay.
Oh, thank you. The time is yours, Ibu Yatri.
The PPT is from the committee.
Oh, 1 second, Ibu. The PPT is already on my screen, Ibu.
Okay.
In our screen.
Can I start now?
Sure. Ibu, time is yours.
Okay. Bismillahirrahmanirrahim. Assalamualaikum. Good afternoon to committee and participants of iBook 2020. Thanks for the opportunity to present our research. Our research title is Antioxidant and Cytotoxicity Activities of Various Extract of Roselle, Hibiscus subderiva from East Java, Indonesia. Me, myself, is Yatri Hafsari, and the rest is my colleague in Research Center for Biotechnology, Indonesian Institute of Science. Next slide, please. The outline of this presentation will be introductions, methods, result and discussion, and conclusions. This is the picture of Roselle or Hibiscus subderiva from Balista that we use in this research. Next slide, please. In Ayurvedic medication, Roselle was used for treatment of hypertension and for the treatment of fever. The red color of Calices itself comes from anthocyanin and acknowledged for potential for antioxidant. The location where the plants cultivated also give different quantity and quality of chemical contents. So in this research, we choose red Roselle, the most common variant that local people use as health treatment or recreational beverages. Dried Calices were extracted with distilled water and various concentrations of ethanol. Then extract were studied its cytotoxicity and antioxidant activities. Next slide, please. Next slide, please. These methods, there are three. Extraction. Red Calices of red Roselle was collected from Indonesia sweetener and fiber crops. Research Institute Malang, East Java, Indonesia. Calices were grounded into powder, then macerated with aqueous. Variation concentration of ethanol 20 until 96%. Ratio of simplicia to solvent was 1:5, and the maceration was conducted 3 times 24 hours. Then filtrate was concentrated with rotary evaporator and dried using freeze dryer. The BSLT assay, the cytotoxic assay, this was conducted based on Meloclean et al. The eggs of Artemia salina were hatched in aerated seawater for 48 hours. Then larvae was put in 10 until 10,000 ppm extract, 3 replicates each concentration. Larvae was obtained, counted after 24 hours. Data was converted using probate analysis program. For the antioxidant, we use ABTS. This assay was conducted according to RE with some modification. ABTS solution was prepared by diluted 90.2 mg of ABTS with 5 milliliter aqueous. The mixed ABTS solution with 88 microliter of 140 millimolar potassium persulfate stored in room temperature for 16 hours. After 16 hours, 5 milliliter mixture ABTS and potassium persulfate was added 155 milliliter of ethanol. This mixture was added to various concentration of extract, incubated for 6 minutes, and measured with spectrophotometry at wavelength 734 nanometer. IC50 value were obtained using regression linear equation. Next slide, please. The result, the first is determination of % yield of extract of Hibiscus subderiva. From this table, we can see that the highest yield showed by distilled water extract. Meanwhile, the lowest showed by 96% of ethanol extract. Distilled water and various concentration of ethanol was used as solvent extract. The purpose of conducting extraction using water and ethanol in order to fulfill requirements from Food and Drug Administration of Indonesia or BPOM. There are numerous methods of extraction such as percolation, diffusion, decoction, maceration, and hot continuous extraction, and parameters can be optimized to gain better yield and biological activity. These methods, maceration itself, was selected in order to isolate volatile and non-volatile compounds. Also, it has more economical value. Next slide, please. For the cytotoxicity, we use BSLT assay. As we know, BSLT assay was a preliminary screening for anti-cancer, and its low cost and rapid screening for investigating compound was preferable than other assay. In this table, we can distinguish that water extract show less toxicity among other extract. Despite value of LC50 of extract overall show less toxicity against larvae of Artemia salina. In exploring lead drug compound, a rapid screening for toxicity activity of plant extract need to be conducted before further step using cancer cell line. SLT has been studied that has positive relationship with numerous tumor cell lines. And LC50 was less than 1,000 ppm. It showed that extract has cytotoxicity activities. Next slide, please. And antioxidant assay use ABTS assay of various extract of Hibiscus subderiva. ABTS assay was selected than other assay because of its solubility in polar and non-polar solvent and stability in broad range of pH. Further, ABTS can decolorize color from extract. In this case, rosal extract was red, so it wouldn't mislead measurement of absorption. In this table, we can see that 80% of ethanol extract had highest antioxidant activity among other extracts. In this assay, we used Trolox as positive control. Trolox is known as water-soluble analog of vitamin E. Next slide, please. So from this research, we can conclude that extract with highest yield was distilled water extract. Highest cytotoxicity were 60 and 80% ethanol extract, and highest antioxidant activity was 80% ethanol extract of Roselle. The acknowledgement, this research was funded by LPDP RISPRO 2020. Next slide. Yeah, thank you very much for listening. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Very interesting topic, Ibu Yatri Hapsari. Okay, since Bapak Sugiarto and his research team is not in this room, so we can skip his turn and we will move to the last presenter in this session. That will be Mbak Andriani. Oh, Mbak Oksi. All right. So the last presenter for this session is Mbak Oksi Vilawardani, who will deliver presentation about detection of drought-tolerant variant in gamma irradiated rice by using RAPD. Mbak Oksi, hello. Can you hear me?
Hello. Yeah.
Hello, Mbak Oksi. Are you ready to give us your presentation?
Yeah, sure. I would like to share my screen.
Oh, sure, you can.
Wait a minute.
Okay, take your time, Mbak Vila Oksi. Time is yours.
Oh, I'm sorry. It's written here has disabled.
Oh, wait. I will talk to the committee and make you as a co-host. All right, you already be a co-host.
Okay, thank you.
Bu Vila Oksiwardani. You're welcome.
Yeah. Okay. Can you see my screen?
Yes, I can see it on my screen.
Oh. Okay, I will start. Imagine if we could develop plants that were resistant to stress. Wouldn't that be a promising solution to the problem of food scarcity? Good afternoon, everyone. My name is Vila Oksiwardani, and I, as the co-author, will present the research under the title of Detection of Drought-Tolerant Variant in Gamma Irradiated Rice by using RABD, or Random Amplified Polymorphic DNA. So let's get started. First, I would like to talk about the background of this research. According to FAO in 2018, rice is the most significant food crop in the world and the main source of nutrition for approximately half of the world's population, as the production is very high. About 167 million hectares are used to rice production, and it's increasing significantly every year. But sadly, almost 130 million hectares land are frequently less. Drug stress is considered to be a loss of water, which leads to somatal closure and limitation of gas exchange. Drug stress characterized by reduction of water content, decreased leaf water potential, and affects turgor pressure. And the result, it can be a It can make photosynthesis inhibition, affects metabolism, so the plants have lower yield or worse plant death. This problem can be overcome by land management or cultivar improvement with assortments and breeding. Land management requires technical adjustment based on field condition, and the variety of field condition make this method less effective. We thought that improvement of drug-resistant cultivars is the right solution because of their stable character in unfavorable environmental conditions. One of the often used method is diversity of the plant based on all of these references. So as in the rice itself, already done by Singik 2012, Kadimi 2016, and Zain 2016. The genetic change from irradiation are very random, so selection of variants are required. Selections are carried out to obtain plants with desired character, and we're looking for rice plants that have resistance to drug stress. The genetic diversity of drug-resistant plants due to irradiation can be confirmed by molecular marker, and in this research, RAPD was chosen because it's suitable for anonymous genome, so we didn't need initial sequencing of the sample plant. It's applicable to limited quantities of DNA, efficient and low cost, and high number of fragments can be formed. And then we'll talk about the methods. First, seed irradiation and germination were performed. Gamma exposure was carried out to two local rice variety, which is Bahbutong and Sigadis, at 4 level of treatment: 0 gray as control plant, 100, 200, and 300 gray. 4 seeds were sterilized and put into a culture bottle that had been given 1.5 grams sterile cotton and sterile water for 7 days. After that, we give them drug stress condition with adding PEG or polyethylene glycol, 6,000 solution at 3 levels, 10, 20, and 30% for 7 days too. Then we calculated percentage growth, termination rate, and simultaneous cultivation 3 replication of Bahbutong variety and 5 replication for Sigadis variety. DNA isolation performed using CTAP method based on Saputro 2018 and used for PCR reaction with RAPD method. Last, germination rate data was statistically analyzed using one-way ANOVA at 5% significant level and used Quentin multiple range test or the MRT for post-hoc test. Data RAPD marker were descriptively analyzed. And then we will talk about the result. This is the result of seeds that grow after germination in culture bottle. You can see that here in figure 1A, B, and C, there were seeds that grew normal, abnormal, and did not grow at all. The spots in figure 1A are similar with the result from Rushdie 2011, so it can be concluded that 1A sprouts grew normal. And these are visuals of the germination results of Sigadis variety seeds with irradiation dose of 0, 100, 200, and 300 gray after 7 days, and the data will be shown in the next slide. First, I would like to explain percentage growth, germination rate, and simultaneous growth. There are common parameters of the plant germination in rice. Percentage growth are calculated with count the sprouts that can survive from the beginning of the treatment of stress until the end of the observation. Germination rate calculated by formula from Sutopo et al. 1988, and simultaneous growth of seed was calculated by count the normal strong sprouts and compared with total seeds analyzed or known with figurative parameter. Based on SNI standard, which used as a normative references, quality requirement and recommendation for rice seeds number 1 year 2015, the percentage growth of rice has a minimum limit of 80%, so that it can be said that the local varieties of Bahbutong at all dose and Sigadis at dose 0 and dose 100 gray are still within SNI standard limits, while for Sigadis variety at dose 200 and 300 gray, the result is below the minimum SNI. But based on statistical analysis, the results show that the increase of irradiated dose gave no significant effect in all plants showed with A alphabet. And these tables are the result of percentage growth and formula height of both irradiated seeds after being treated with drug stress. All Bahbutong plant samples were able to withstand drug stress condition, which given 30% PEG, but Sigadis varieties could not survive. You can see that here in 30% lines all showed 0. This indicates that the irradiation Sigadis. Based on the results of statistical analysis, which listed in the table indicated by letter A, it can also be seen that there is no significant difference between giving PEG 10% and 20% with control plants. In this study, three RABD primers were used to characterize the genome of irradiated rice plants that were able to survive in 10, 20, and 30 drop conditions. The polymorphic information content, or PIC, shows the informative level of primers, which means that the primers are able to attach to sites that experience an alteration due to mutation generated by irradiation process and amplify the fragment so that a polymorphism phenomenon occurs. The PIC value is divided into three classes, which is low, less than 0.25, medium, between 0.25 until 0.5, and high, which is greater than 0.5. The total number of loci detected from all three RABD primers in both variants, which is Sigaris and Bahbutong, was 49 with 44 polymorphism loci. The result showed that the irradiation caused change in nitrogen-based composition of the mutant lines detected by all primers, which range medium or PIC value are between 0.25 until 0.5, and OP810 in Bahbutong varieties has the highest polymorphic band with a PIC value of 0.4881. Last is the conclusion. So far from this research, we know that the best irradiation dose for drop stress in rice is 100 gray, which showed best performance in percentage growth and pulmolar heat parameters, almost same as the control plan in drop stress. Second, we can conclude that alteration in nitrogen base is source of no variance in mutant lines. Furthermore, all chosen primers showed a high rate of polymorphism and expected that the genetic change occur. That's all for me. Thank you for all of the person contribute in this research, which is member of Plant-Based Science and Technology Laboratory, RSKI Unair, Baitkabi, and Batan. And thank you for your attention. I'm sorry if I make any mistakes. Good afternoon.
Good afternoon, Ibu Villa Oksi. Thank you for your pleasing presentation. Now we will move to the last presenter for this session. It will be Ibu Andriani. So Ibu Andriani will deliver presentation about the genetic diversity and morphological responses of Capsicum annuum varieties under aluminum stress. Halo, Mbak Andriani.
May I share my...
Are you ready to deliver your presentation?
Yes.
Okay. So just to share screen for us. Okay, Ibu Andriani, so time is yours. You can start your presentation.
Thank you very much. My name is Andriani. Here I want to present about the genetic diversity and morphological response of Capsicum annum varieties under aluminum stress. Capsicum annum is one of the most widely cultivated vegetable crops in the world. The fruit contain various beneficial metabolites like capsaicin and capsitol, so it can use for food industry and health industry. To increase the productivity of Capsicum annum, it can by pesticide or by fertilizer. But one of the negative impact is the accumulation of heavy metals, including aluminum stress. Aluminium will absorb by the plant from the root, and then it will make rust production and dysfunction of mitochondria, so it's can toxic in the plant. Any very much variety in Indonesia, and the one there is a Chico, Tanjung Dua, Lingga, and Lembang Satu. But information regarding the intraspecies genetic diversity of Sili is still limited. So need method for to know the intraspecific genetic diversity. One of the method is ESSR. ESSR are known as RAMS, Random Amplified Microsatellite. Microsatellite are regions in the genome that consist of short DNA motif, usually 2 until 5 long nucleotides that are repeated several times in succession. This research purpose is to determine the genetic diversity of several varieties of psylli based on ESSR marker and to determine the morphological response, plant height, and root length of several psylli varieties to the stress of aluminum heavy metals. And the methodology, the first is the psylli are planted and then growth until to leaf, and then analysis of genetic variation using ESSR markers with 5 ESSR primers. This is ISO2, primer 5, primer 9, ESSR 8 to 5, ESSR 811. And then we give the aluminum stress 32 days. And then we see the morphology response. This is a plant height and root length. And this is the result. The first is analysis of genetic variation using ESSR marker. We get several polymorphism band, and this is the example of the polymorphic band. And the band size is we get 200 until 1,700 base pair. And then we we measure the PEC value. PEC value is often used to measure the informativeness of genetic marker for linkage analysis. We get for primer 9 is high informative, for primer ISO2 is less informative, for ESSR 825 and ESSR 811 is less informative, and primer 5 is quite informative. And then we analysis in UPGMA, and the result is Lembang 1 is closely related with Lingga with similarity is 0.5, and Tanjung 2 is closely related with Chico with similarity is 0.6. And then this is the heavy metal effect on the plant height. It shows the decrease in plant height compared with control. Why is decreased? It might be because aluminum can reduce the absorption of nutrients such as phosphate. Phosphate that decrease because the heavy metal Al will react with phosphate, so it will bind the anion and precipitates it as a reaction Al phosphate by the reaction. And then
the group one, the diptera start from the fresh.
For the potassium is decreased because maybe the ion Al3. Ion Al3 may be bind with the phospholipid heads of the plasma membrane, which is in turn interact with protein lipids and modifying the transporter activity. And then the nutrient that reduce, it will cause the reduction in plant growth and biomass. And then for heavy metal root length, in the ANOVA, it shows the influence of the varieties of Capsicum annum and stress concentration of heavy metal Al on the root length. This is the picture that shows that Lingga 0 ppm and Lingga 100 ppm. In 100 ppm, there is a decrease of the root length because maybe the localization of Al accumulation lies in initial centimeter from the root tip, and the accumulation of Al in the apex of the root will cause cell death, thereby inhibiting root elongation. Why it's become maybe the aluminum will enhancement of the roots, and it will starting the PCD in the root. So it's make the reduction in mitotic activity is recorded. The root is determined by the rate of the cell division in the meristematic zone and cell expansion in the elongation zone. So it will make the decrease in the root length. And the conclusion from this research, the first is the ESSR analysis using 5 primers. So the PEC value between the 0 in primer 8 to 5 to 0.6 in primer 9. UPGMA shows that the Lembang 1 is closely related to the Lingga, and the Tanjung 2 is closely related to Chico. And the second is the morphological response, so that the interaction between the Capsicum annum varieties and variation in the concentration of heavy metal Al had an effect on the root length. Al stress on the Lingga, Lembang 1, Tanjung 2, and Chico showed a decrease in the plant height compared to the control. Okay, thank you very much for your nice attention.
Thank you, Mbak Andriani, for letting us know about what is the effect of aluminium metal on Capsicum annum growth and the root length of Capsicum annum. So that was our last presenter. And now we will go to the Q&A session. Any queries for our presenter? Does anyone has a question for our presenter?
Can I ask a question?
Sure, Professor Setia Marga.
Okay. Everybody did very, very well, but one thing, there is one question I would like to ask to them. I don't remember. It's about the irradiated rice.
Irradiated rice. Oh, and the fourth presenter, Ibu Villa
I think
Oksi.
so, yeah. Right. So my question is that after you are trying to basically find useful mutations, induce useful mutations in wild rice. In the rice. Using irradiation, I think, will introduce only random mutations, and you will have to do a lot of efforts in order to get a very, very good result. Why don't you try to use say like something like CRISPR or something like that in order to target specific mutations to the gene that you would like to change? I think it will be like more it will be faster, it will be
I think Prof. Davin is lagging.
Yeah, he has a connection issue. So we wait for his next question. I think he is stuck because the connection issue.
Yeah.
All right, you can answer, Mbak Villa Oksi.
Oh, okay. Thank you so much, Prof. Davin, but I know he can't hear me. But actually, the irradiation is the trial for our research. At that time, we didn't know CRISPR yet. You know that in Indonesia, it's likely what do you call? We didn't.
It's kind of a sophisticated method, maybe in.
Yeah, it's very new method, and we didn't have them at the time when we perform this research. Yeah, if we can, I would like to use that method too, but it's so expensive and yeah, Thank you.
Oh, thank you for your answer. How about it, Prof. Setia Marga? I think he is no longer in this room because I just searched his name, but I couldn't find anything about his name. So can I ask the last question for presenter number 2 for the second question? For the second presenter, Ibu Suni, I just curious about why did you choose chicken manure as additional nutrition in your pot? Halo Ibu Suni. Halo. Welcome back, Prof. Setia Marga.
Yeah, I
Seems like
actually,
you got an internet issue in Japan.
yeah.
Yeah, Prof.
But
Actually,
my
I would like
I
to.
was able to listen to the answer through my other computer, but I was unable to reply back because I don't have any camera and microphone for my other computer. But yeah, thank you.
Okay, Prof. Thank
Okay.
you for your suggestion. I think I would like to replay my answer. Is it okay, Mr. Andri?
Sure.
Yeah,
I don't know.
Yeah. Actually, at the time, the irradiation method is our trial and error. So here in Indonesia, at the time we didn't have CRISPR in ITS. I mean, and we use the irradiation method to what do you call to induce the mutation of rice. So because of that, and you know that CRISPR is sophisticated and very expensive method. So well, yeah. Thank you.
Besides CRISPR, there is actually other method that you will allow you to induce mutation more specifically. For example, you can build a construct with a plasmid, put it in the E. coli, and then you can try to induce it in the transposons and stuff like that. I think it's like much cheaper and faster rather than irradiation, because in my opinion, because I don't irradiation is more expensive because you have to have the access to the facility allowing you to use radioactive materials.
Actually, the issue for us because we just send the sample to Batan. That's yeah. So
Badan Petasan Nasional.
Yeah. And for the plasmid reconstruction, at the time too, or first, sorry, but in ITS we didn't have it yet at that time. But maybe we will do that in the next project. Thank you for your suggestion, Prof.
Thank you very much.
Thank you very much, Prof. Setia Marga, for your valuable suggestion to Villa Oxy for her next study maybe. So later on we will have a break for 10 minutes. So we will join again in this room around 13:47. So see you in the next 10 minutes.
Okay.